Hi, guys. So today we're going to be discussing an interesting topic from cell pathology, which is intracellular accumulations. So to begin with, how do cells respond to stress? So cells have to maintain a state of structural and functional integrity to be alive. And if factors around the cell changes, these can be internal factors or external factors, we call these changes as stresses for the cell because now the cell has to adapt in order to survive in this scenario. So, what determines how the cell responds to stress? So, this depends on how long the stress acts, how strong the stress is, and the type of the stress. And it's not any individual factor alone that determines how the cell reacts, but a combination of these factors. For example, if a stress acts for a very short time, maybe it's just a momentary lack of oxygen, then the cell might be able to adjust to this. But if, say, there's no oxygen for, say, 10 minutes, 15 minutes, then the cells would likely die. Also, we can talk about stresses such as electricity. If you get electrocuted, this stress might act for just a moment, maybe a few milliseconds, but this is a very severe kind of stress. So it will also result in cell death. So this is why we could say that in general, we have a combination of factors which decide how the cell responds to stress. So let's talk about the different types of stresses and how cells would respond to these. Why I'm getting into all these for intracellular accumulations, you'll understand in a moment. So, if the nature of the stress is such that it's quite physiological, maybe small changes in pH or temperature, or even the functional load on the cell increases, then the cell could just simply adapt. Just think about it like, say, you're in medical school. So if you have a small amount of stress, such as, say, you have a test next week, you would probably not give up med school. You'd rather you just start working harder if your load increases, right? So it's similar to this. Uh, if you have hypoxia uh, or some sort of physical, chemical, or biological stress, depending on how severe and how long the stress acts, if it's just quick and it's mild, the cell will get injured, but temporarily, and it's reversible. But if the stress is long lasting and progressive, the cell is unable to adapt to this. It's just like the same thing. Let's take the example of med school, right? So if you have one test a week, then you can probably try to study harder and adapt to this. If one week suddenly you have three tests, then you're really stressed out, you work really hard, but since it's just for that one week, you might be okay with it. And once it's done, you're feeling better again, you adapt and you return to your original state, you return to homeostasis. But if your stress continuously increases, so say this week you have two tests, next week you have three tests, and then you have four tests, and then you have two tests a day, and this goes on for, say, a period of like 4 months, then this is some stress that you can't handle and the damage is irreversible and you'll probably give up and quit med school. It's the same thing with cells. So if the stress is too severe and progressive, then the injury is irreversible. So you have some other types of stresses. So if the cell, due to some sort of genetic anomalies or due to some metabolic reasons, it could be nutritional, then the cell would react with a kind of regressive change known as an intracellular accumulation. It can also undergo pathological calcification. We will learn what these things mean very soon. If there's long-term injury on a cell, but it's not something that's not exactly non-physiological. Say you have a lot of increased functional load for a very long time, but it's just a mild amount of increase, but the damage accumulates over years and years and years. Think about a machine running, right? Like a car. When you use it every time, there's regular wear and tear. So eventually, after some time, your car is gonna reduce in quality. And at one point of time, that's kind of the end of the lifespan of your car. It's the exact same thing. It's the same way cells respond to stress. So we have four ways. So one is they adapt, either they or then they can get injured reversibly. If it's really severe, then the injury is irreversible. If the kind of stress is from a genetic or metabolic perspective, then they can accumulate substances within them. And if you have long-term injury, which is sublethal, then this is known as aging. Okay. So what are intracellular accumulations? So we learned that if there's some sort of metabolic or genetic damage or stress, then cells accumulate substances. So intracellular accumulations are substances which are either formed within the cell, which is what is meant by endogenous. So something that either the cell is making or it's within the cell, maybe created by another cell or maybe even the extracellular matrix. Uh, or it could be exogenous substances such as maybe dust, pollen, or even microorganisms. So anything that is of endogenous or exogenous origin, which can accumulate in the cytoplasm or any of the other cell organelles, are known as intracellular accumulations. These can either be completely harmless or sometimes they can cause further injury. For example, maybe from histology, you guys remember that you have certain cells that never divide, like say cardiomyocytes and neurons. So they accumulate a substance known as lipofuscin, which is from lipid peroxidation reactions. And in general, you can think of it as waste products of metabolism. And the cell doesn't really know what to do with this. So lipofuscin accumulates over years, but this isn't necessarily damaging. But for instance, say if your cell phagocytoses something it can't consume, or it's unable to degrade a really big molecule in the cell, this will actually cause further stress because it increases the energy demand and the cell gets further injured and may die. So intracellular accumulations are reversible. It doesn't have to be permanent. So it's not cell death. So if you're having intracellular accumulations, it's still in a reversible stage. But again, it depends on the disease and the metabolite being accumulated. So now we're going to talk about how these substances get into the cell. So either it could be a normal substance, such as, say, lipids or carbohydrates, which are not being removed properly from the cell. It could be due to maybe low energy production from the mitochondria. So, for example, say water. So, if your mitochondria is failing, say due to some sort of toxin, then you can't really regulate your ion flows and your sodium potassium pumps fail. And then you can't really, maintain your ion equilibrium. So, you have water starting to accumulate within cells. So, this is basically inadequate removal of a normal substance, right? Water is normal, water is physiological. But if you can't remove it, it accumulates and it is damaging. Next, you could have something like defects in protein folding. So proteins should have a certain tertiary or quaternary structure, and if there's some, say, small point mutations in DNA and there's a change in codon, it could switch some of these amino acids up and they're not able to fold properly. And because of that, other molecules which are supposed to aid them in moving around in and out of the cell, or maybe, even for vacuole formation or peroxisome formation. So basically, your endoplasmic reticulum and all your systems, your organelles are not able to coordinate because of the wrong protein. So in such a circumstance, this protein can't be excreted, can't be degraded, so you can't do anything with it. So the cell just has to keep it. It accumulates. The third example is kind of coherent with the second one we were talking about. So the third mechanism is when the cell cannot get rid of a component, say due to the lack of an enzyme. So say there's some mutation in the DNA or some sort of damage in the cell and you have loss of an enzyme. And because you don't have an enzyme, you really can't break down that substance. And if that substance is too big to leave the cell, it has no choice but to stay within the cell. So that's the next mechanism. And finally, we could have external substances which are accumulating in the cell. Suppose, say, you have dust. Maybe if you guys remember sections of the alveoli from histology, you could remember there were some black particles in the cell. This was dust. So this is also a type of intracellular accumulation of an exogenous material. Okay, so now quite an easy question. What can accumulate? Pretty much anything. So water is the first one. This is one thing I think further along the line people tend to forget. Everyone remembers lipids, proteins, carbohydrates, pigments, but people often forget water. Even water accumulating in excess is damaging to the cell. So it's important to mention this. Then, other than your basic biomolecules, you can also have foreign bodies accumulating. You can have crystals and microorganisms. Now, first, water. Before I even speak about water, I want to say that in today's video, I'm not going to be able to cover all the different topics, but I'm going to try my best, at least to start with pigments. I won't be covering all types of proteins, but we'll get to that as and when we get through the video. Okay, so water. So when water accumulates in the cell, this is known as hydropic degeneration. It's a type of regressive change. And if you're wondering what regressive change means, it's just something that's damaging to the cell or detrimental or backward, right? And this happens due to energy deficiency, as I touched upon earlier. How this happens is when the mitochondria fails due to some external condition or internal condition. Say it could be toxins, it could be sepsis, it could be inadequate oxygen. So this causes mitochondria to fail. What happens if the mitochondria fails? You don't have ATP. If you don't have ATP, all your pumps fail and you can't regulate ions. So if you're not able to regulate the ions, you're not able to coordinate the movement of water in and out of the cell. So this is called hydropic degeneration. So other than toxins, anoxia, and sepsis, certain conditions which are supposed to even starvation or autolysis can cause mitochondrial failure. But if you want more examples, they're there in Professor Dushkova's lecture. This is just an overview. You can also have lipids accumulating. So this is known as steatosis. What is steatosis? So by the way, in pathology, guys, you need, whenever you're talking about anything, it's good to start with a strong definition of what you want to say and then give examples. So, okay, so we want to talk about steatosis. What is the definition of steatosis? It's when lipids and triglycerides and different types of maybe even cholesterol accumulates abnormally within the cell. So this is steatosis, and this can be classified based on the mechanism of how steatosis happens. So I think the simplest way to think about it is okay, even before we look at the options. So if you're having an excessive calorie intake of lipids, there's too much of it, and you're eating more than your body can digest, then you're going to have your cells are going to accumulate lipids. If there's a system which is failing in the transport of lipids, maybe again due to some sort of enzyme deficiencies, you're not really able to degrade the lipids, then you have steatosis. Also, sometimes a cell becomes sick, so it's not able to perform normal metabolism properly. So under this scenario, still you can accumulate lipids. So these are a few examples, and the proper terms for these are as follows. So saginative steatosis is when you have an excessive calorie intake. So in the case of hyperlipidemia, you would see this. Sometimes you have steatosis of like you have transportative steatosis. This happens during, say, starvation. So in starvation, what happens is you have all your like reserves being broken down to be able to obtain energy to make up for the lack of energy, right? So if you don't have enough glucose in your body, so because we all know that carbohydrates are our first line. So as soon as you stop eating, first your body tries to use up the carbohydrates, and then for gluconeogenesis, you need to mobilize free fatty acids. But if the transport is not working out properly, for example, yeah, in this scenario, say the patient is extremely starved, then you would actually have the cells reacting absurdly and accumulating these lipids. Then you could have resorptive. So this is an interesting one because, so for example, you have hyperlipidemia, the same scenario we spoke about in the first time, right? And now, because the lipids are so high in the body, the immune system is trying to clear it out. So, which is what your macrophages are doing. So, what your macrophages do is they go and they eat these lipids. But once it's in the macrophages, they don't know what to do with it either. So, it's stuck in the macrophages and they form something known as foamy cells. This is something known as resorptive steatosis. So, just a bit of a, I think, a bit of extra information. You will do this in summer semester, but so when macrophages are cleaning up all the lipids, right? If this happens for a prolonged period of time, this is kind of your precursor for forming atherosclerotic plaques. So essentially, you can think of stagnative steatosis becoming resorptive steatosis. So there's no like distinct lines. I think it's just good to know the different mechanisms, how lipids accumulate, but there's no distinct categories because it kind of goes hand in hand. So if you have excessive calories, your body is also trying to clean up these lipids faster, but they can't do anything, and yeah, it accumulates. So retentive is rather genetic. So, say you have a category of diseases known as lysosomal storage diseases. And basically, what happens in these diseases is there's a lack of a certain enzyme, and you can't break down a certain compound. And because you can't break it down, it's retained within the cell. This is also seen for other biomolecules such as proteins and glycogen, which we will go through soon. Okay. So, just a little bit of additional information. Anything that is affecting the general metabolic pathways of this particular biomolecule, in this case, lipids, right? So, uh, lipid mobilization, transport, storage is done by a variety of organelles in the cell. So, for transport of fatty acids through the mitochondrial membrane, you have the Carnitine shuttle system. If there's a disorder which affects it, if there's a toxin which targets the system, then automatically, because it fails and the lipids cannot be sent into the mitochondria, they have no choice but to accumulate elsewhere in the cell. So, this is an example of a mitochondrial disease or toxin against mitochondria leading to steatosis. Similarly, you could have a situation with lysosomes, where so we know lysosomes are the storage organelles, right? So if there is a dysfunction of say certain enzyme and you don't know what to do with the lipids, you have to accumulate it somewhere, right? So you do it in the organelles lysosome. So you can have congenital dysfunction, which is called lipidoses, and if there's some other external factor which is affecting your lysosomes, you call it acquired dysfunction, lipophages. You also have peroxisomes. They're really important in lipid metabolism, and some disorders with these could lead to steatosis. And also, lipoproteinemias are the main mechanism of steatosis. As we discussed earlier, it was with the saginative type. Okay, so how does steatosis look? So, how do I look at an organ and decipher that it has accumulated fat? Oh, also, a simple way to look at steatosis, a simple name for it is just fatty change, right? So, how do I know that an organ has undergone gone fatty change? So, in this case, we see the normal healthy liver, and you can see the second picture, which shows fatty liver. Wait, let me see if I can point. Yeah, and I hope you guys can see it. Right. So in the case of moment. Okay. So the normal liver, the parenchyma looks fine. The liver is small in size, just as it should be. But in fatty liver, what's happening is the liver is swelling up. It's becoming more yellowish. And if you're able to touch this liver in person, you would see that it's very soft and greasy. Just think about it like oil, right? Lipids, oil. How is oil? It's greasy, it's soft, it's smooth. So this is exactly how the gross appearance of steatosis would be. And when you look at it microscopically, which you just need to know in terms of, you just need to know to describe it. You don't actually have to look at a microscope and be able to identify steatosis. It doesn't matter. Just know that you have microvascular or macrovascular steatosis. Okay, so which conditions or which diseases do we see steatosis in? So we can see it in alcoholism. We can see it in non-alcoholic fatty liver disease. We can see it in cholesterolosis of the gallbladder. We can also see it in starting stages of atherosclerosis, which is the foamy cells I discussed earlier. And yeah, also in complex atherosclerotic plaques. Another interesting condition where we see steatosis is myocardial steatosis. This is called tiger heart or tabby heart. And how this happens is the stress in the situation, because remember how I specified? So cell is normal, it needs to encounter stress, and the reaction to the stress is accumulating some substance, right? So let's trace it backwards. So for the heart, the stress here is chronic ischemia, or not ischemia, I would say chronic hypoxia. It could also be ischemia, but okay, for the sake of a discussion, let's say we have chronic hypoxia of the heart. And when the heart is having insufficient oxygen for a very long time, then this is a stress on the heart. And because of this stress, due to less oxygen, it's affecting probably the mitochondria. And because the mitochondria is getting affected, you know, you're having some disorder with the fatty acid mobilization, and your heart is starting to accumulate these fats, and it has a stripe-like appearance. Just think of how the heart looks, whatever you've seen, and imagine like yellowish, greasy stripes all over the heart. So this is myocardial steatosis or tiger heart. Okay, this is what you call as your strawberry gallbladder, and it happens when you see all these different yellowish substances, yellowish granules. So this is on the surface and in every cell. So basically, the cells are accumulating cholesterol over a long period of time due to some sort of dysfunction in the cholesterol production system. And by the way, when I say lipid accumulation, I mean any lipid. So triglycerides, fatty acids, cholesterol, it doesn't matter. Okay. Um, right. So here it's basically a gallbladder with accumulation of cholesterol, and this is called the strawberry gallbladder. Now we're going to talk about inborn errors of lipid metabolism. They're known as lipidoses. They're really rare, and they come under a category of lysosomal storage diseases. So the lipids can be stored within different types of cells, and you don't really need to know the exact symptoms of any of these diseases in detail, but it's good to know in which cells, particularly, that these lipids accumulate. And you might ask why only in certain cells. I will get to that soon. But anyways, for now, know that lipids can accumulate within macrophages, neurons, or pretty much generally everywhere, depending on the type of disease. So the first one we are going to speak about is Gaucher's disease. It is caused by the deficiency of beta glucosidase enzyme. And because of this, you're not able to degrade a substance known as glucocerebrosidase. And because this lipid is not being degraded, it gets accumulated in the cell. And where this enzyme is usually supposed to be found is in the brain, right? And the neurons. So because there's a deficiency of this enzyme in the neurons, the brain gets affected. So similarly, there's a few other disease. I don't want to go into like the detail. Simply, it's good to learn this table, but and I highly recommend it. You should learn this table. But the thing is, there's really no explaining to do. You need to know the name of the disease, the deficiency of the enzyme, and which lipid is accumulated. And just so that you get an idea, know that for Gaucher's and Tay-Sachs and for Niemann-Pick disease, the main tissue involved is your brain. So the brain is where the lipids are getting accumulated. So naturally, the symptoms would be psychomotor retardation and, maybe low intellect, things like that. So everything that has to do with, reduced cognitive function would be the symptoms you would see in these diseases. And most of these diseases are eventually lethal, but yeah, so it's good to know which organ is involved primarily, and it's the brain. If you're ever asked in the exam which other organ is involved, in almost all lysosomal storage diseases, you need to know that both the liver and the spleen accumulate these substances. So one of the symptoms you would see in almost all LSDs is basically hepatosplenomegaly. So any kind of LSD, so it could be lipidoses, it could be any kind of like a peroxisomal disorder, anything, right? Okay, so next we're gonna go into protein metabolism. So proteins can accumulate either within the cell or outside the cell. So since we're talking about intracellular accumulation, I'm not really gonna touch upon amyloidosis because we have a separate, like, amyloidosis is deserving of its own video, so I think someone's doing that. But anyways, nevertheless, we're gonna speak about two other types of extracellular accumulation of protein. But our question right now, we're going to be discussing mainly the intracellular accumulation of proteins, right? So you need to know the mechanisms. So how does protein accumulate in the cell? It's kind of a similar mechanism to say lipids. So suppose say there's some sort of defect in the transport system, right? That could cause the protein to accumulate. If there is some sort of misfolding, or it could even be the like inadequate removal of say normal protein due to the lack of an enzyme or something of that sort. But you have something interesting to note, which is known as hyaline change, which is what we're going to be discussing soon. So I'll go through that. Okay, so we're going to be speaking about hyaline change and fibrinoid change. Okay, so fibrinoid change or fibrinoid dystrophy is a type of reversible change in collagen. Even hyaline change is a change to collagen, but both of them have different like causes, different etiologies, right? So fibrinoid dystrophy happens due to increased pressure. And one of the good examples of fibrinoid dystrophy is what you see in fibrinoid necrosis seen in blood vessels. It happens due to the circulation of antigen antibody complexes, and it's a type 3 hypersensitivity reaction. So, this kind of change to collagen would decrease the firmness of of the protein and increase the permeability. So I will try to show you how the picture of this change looks. Yeah. So here, as you can see, this is the blood vessel, and you can see a pink homogeneous substance, which is deposited in the vessel wall. Here you see the red blood cells, right? So, I don't know what I was gonna say. Right. So basically, anyways, yeah. So hyaline chain, fibrinoid. Oh yeah, I remember. So the reason this is called fibrinoid change is because this stain a lot like fibrin. Be careful that I've seen a few resources say that fibrinoid change is due to deposition of fibrin. This is not true. Fibrin is a different component. Fibrinoid is a term used to describe staining similar to fibrin, but fibrinoid dystrophy is due to the change of collagen, not due to fibrin, right? They're completely different proteins, so don't mix it, right? Now we have hyaline change. So, oh, also, fibrinoid change can be reversible, but this isn't the case with hyaline change. So, hyaline change is a stable change and it can occur intracellularly or extracellularly. And this is also a change to collagen fibers. But the difference is for hyaline change, it happens due to the thickening of collagen. But for the fibrinoid change, it's due to the interaction of it's due to the interaction of collagen with plasma proteins to form this fibrinoid material, right? So in hyaline change, the thickness increases, and because the thickness increases, the elasticity of collagen decreases. So this messes up the quality of the extracellular matrix and the connective tissue. Causes could be due to organ ischemia and sorry, not causes. So what this causes is organ ischemia because think of it, right? So you have extracellular matrix of say cardiac muscle, right? If the connective tissue is increasing in amount, then it's really hard to send oxygen to the cells because it needs to diffuse across the cell, right? So if the ECM is really thick, then the oxygen needs to travel across a further distance. So this will eventually reduce organ function. So these are some examples of hyaline change. So intracellular hyaline change is seen in Mallory bodies, which is basically a type of a cytoskeletal accumulation of protein. And then Russell bodies are something unique, which is seen in certain types of blood cancers, such as multiple myeloma. And Crooks hyaline is a type of change you will see in the adrenal cortex and also the pituitary gland. You'll see these kinds of cells. I will show you how they look, but this is more important for summer semester more than the winter semester. Anyways, you also have extracellular hyaline change, and where you can see this often is in scar tissue and yeah, you can also see this in certain types of vasculitis, which you'll learn what is vasculitis in summer semester, but it's basically inflammation of the vessels due to, it could be, say, due to a type 3 hypersensitivity reaction. So, yeah, you don't have to get into too much detail. It might be confusing for you. So just focus on intracellular hyaline change now. More of the other things will make sense in summer. Okay, so in the first picture, you're basically seeing, so this is yeah, so this is cardiac muscle, and in this cardiac muscle, you're able to see hyaline change in these places. Uh, this is a picture of Mallory's hyaline change. So basically, you see these pink homogeneous substances in the cells over here, different places, right? So this is your Mallory hyaline change. This one is your Crookes hyaline. And yeah, this is a picture of the adrenal cortex. This is something you call a Mott cell. These different small vacuolar looking things, these are basically what you call as your Russell bodies. Okay. So these are your Russell bodies in the cell, in a white blood cell. Okay. Right. Now we're going to go through diseases of glycogen metabolism. Okay. So, a lot of conditions could impair how glucose or any carbohydrate is metabolized. So that we know that the two most important hormones for maintaining and regulating uh, glycogen is insulin and glucagon. So anything that is affecting the levels of insulin and glucagon in the body is gonna result in a disease of glycogen metabolism. So the easiest ones to remember are diabetes type 1, type 2, latent autoimmune diabetes in adults. And there's also something known as mature onset of diabetes in youth. It's called MODY, right? You don't really need to know the details, but just know that such conditions exist. Then you can have certain tumors of um, the insulin and glucagon secreting cells. So this is called an insulinoma or a glucagonoma. So this essentially either increases or decreases the secretion of this hormone and dysregulates metabolism. You can also have conditions such as obesity and metabolic syndrome, which affect glycogen. But also you have certain normal, not normal, sorry, you have certain pathological conditions such as clear cell tumors, which leads to the accumulation of glycogen in organs such as the kidney, breast, ovaries, etc. If there's an enzyme that is missing in glycogen metabolism, then you have something known as glycogenosis. So you have a massive list of diseases which are caused due to enzyme deficiencies involved in glycogen regulation. So it's good way to it's a good way to look at which organs are being affected. So let's go through that. So I try to make a table for you to simplify what is happening in these different diseases. So if the liver is the primary organ being affected, we call it as hepatic glycogenosis. If it's the muscle, then we have myopathic glycogenosis. If it's the nerve, then it's neuropathic, or you could have generalized glycogenosis. You need to know which enzyme is deficient and the severity of the disease. So usually glycogen storage diseases have two names. One is just the numerical naming, so suppose say a GSD1, but you also have the name of a scientist. So this is called von Guericke's disease, right? So I'm not sure if you need to know both, but I think it's a good idea to learn them. And this table is pretty much, you know, uh, it's conclusive of everything you need to know. So just go through this. Okay. Then one of the major diseases of glycogen metabolism is diabetes mellitus, but I'm not really going to touch upon this question, although they ask from it. When you're talking about glycogenosis, they expect you to talk about diabetes, but diabetes has its own question reserved for the summer semester. So I don't want to be redundant, so I'm going to skip it. But for now, we'll just keep a simple definition. that diabetes mellitus is a group of chronic metabolic disorders characterized by hyperglycemia. So this is what's common between um, all the different types of diabetes, right? And it occurs due to either a defective response to insulin or defective secretion of insulin, right? So, pigments. Now, so far, we discussed that water can accumulate, lipids can accumulate, proteins can accumulate, and under proteins, we discussed the accumulation of hyaline, and we also discussed the accumulation of fibrinoid. We've not yet done amyloid accumulation, and it has a separate video. But now, let's dive into accumulation of pigments. So, what are pigments? Pigments are colored substances in the environment and or in the human body, right? So pigments are like so for our understanding, we can classify them into two general types: endogenous or exogenous. So endogenous pigments are produced within the own body, while exogenous pigments are from outside, from nature, from the environment, right? So endogenous pigments are further classified as autogenous and hematogenous. Autogenous pigments are produced by uh, as the products of metabolism. So, for example, say melanin, right? The primary pigment in skin and our retina. This pigment is a product of metabolism. You can also have some other pigments which are derived from blood, primarily from the breakdown of hemoglobin, such as bilirubin, biliverdin, and so on. And yeah, so exogenous pigments enter the body via respiration, GIT, or traumatic lesions. So think about it, right? So you can inhale something that's colored. You can eat something that's colored. For example, say if you eat a lot of carrots, you would see that your GIT starts to stain orange after a while. If you eat a lot of beet, then your entire GIT stains a little reddish. So these are exogenous pigments, right? And for respiratory route, it could simply be dust that you're inhaling. Or traumatic lesions, the best thing to think of is tattoos. So you're deliberately pricking the skin and disturbing the layer to inject some inert inks into the skin. So uh, it's an exogenous pigment in the skin. Okay. Yes, so examples, we already spoke about this briefly. So let's speak about the first endogenous pigment. And to be specific, it's an autogenous pigment, so melanin. So when you're speaking about it in the final exam, you need to speak about how this pigment is produced in the body, its functions, and what pathologies can happen with, say, maybe excess of the pigment or how it looks, some general idea. So it's about its accumulation, basically, right? So production, function, and accumulation of pigments, because pigments exist in the body for a reason. So melanin is of two major types, eumelanin and pheomelanin. Eumelanin is brownish black in color, while pheomelanin is yellowish red. It's yellowish red because it's rich in sulfur content. What is the function of melanin? So it's very important in protecting cells from the harmful effects of the sun and the UV rays. It's also responsible for maintaining visual acuity. So the pigment in our eyes is what helps us to see better, right? Then it also has a major detoxification function because it exchanges ions and gets rid of toxins. What disorders do we have of melanin pigmentation? So either you can have overpigmentation. So in the case of, say, so you have hyperpigmentation and hypopigmentation. So for hyperpigmentation, if it's all over the body due to, say, some sort of systemic disease, so or it could be the disease of a certain organelle, sorry, not organelle, so of a certain organ, then you can have hyperpigmentation. For example, Addison's disease is a disease of adrenal insufficiency. So when you have adrenal insufficiency, your pituitary gland is trying to stimulate your adrenal gland to produce more hormones, but this isn't happening. So it's like a positive feedback loop. So bad function of the adrenal gland is leading the pituitary gland to function more. And a hormone which is stimulating the adrenal gland is your ACTH. And another byproduct of the cleaving of ACTH is actually it's alpha MSH, and it's basically a melanocyte stimulating hormone. So if you have Addison's disease, which is affecting the adrenal gland, you also have pigmentation generalized all over your body. So some other examples of hyperpigmentation are local, so freckles, nevi, chloasma, and melanoma. I will get into details of what these are. But you also have disorders of hyperpigmentation. It can be generalized, such as complete albinism or partial albinism, or it can be local, vitiligo and leukoderma. So first picture we see here is a patient. It's a little girl with albinism. Here, the patient has vitiligo. So what vitiligo is it's an autoimmune destruction of melanocytes. So it's causing depigmentation. You can see that it's quite well circumscribed, if that makes sense, if the depigmentation is quite well circumcised. In this situation, we have a patient with leukoderma. So understand that leukoderma is always post-inflammatory, while vitiligo is autoimmune. A lot of sources tend to mix these up, but our university expects them to treat it as two different conditions. And they say that in the case of leukoderma, the delineation of the hypopigmentation is kind of it's not very sharp. So here you can see that the borders aren't that sharp while when the skin is getting depigmented, but here it's kind of well circumscribed. You can exactly pinpoint like the regions as to where there's less pigment, right? So the first picture we see here is from Addison's disease. We have some pigmentation in the oral mucosa and the tongue. Uh, this is chloasma. So chloasma is a type of hyperpigmentation that occurs during pregnancy. It can happen on the skin of the face, the nipples, and the genitalia. And this is a more dangerous condition. It's actually a melanoma. So it's basically a type of neoplasia. I don't want to use the word neoplasia yet, but it's the most appropriate word to use. You'll learn about it later when you're doing oncology. But basically, to put it simply, melanoma is a type of cancer of melanocytes. Right. Uh, so basically, one second. Right. So you also have some rather kind of physiological conditions where melanin can be deposited in excess. So among these are nevi and freckles. So freckles are also sometimes, sorry about, yeah, okay. So, so you have some rather physiological, not always physiological, but there's a thin line, right? So you have something known as nevi, or often your birthmarks. So you have something known as your intradermal nevi, which is basically, you know, this kind of lesion that you see here, which is normal on your skin. So a lot of these lesions are due to activity of melanocytes, and depending on how they're localized, we classify them differently. So basically, freckles occur when there's no increase in the number of melanocytes, but certain cells have an increased amount of pigment. Lentigo occurs when there's an increased number of cells, but not necessarily an increased amount of pigment. I mean, obviously, if this number of cells increase, pigment is going to increase, but not for the individual cells. You also have some other types of nevi, such as the junctional nevi, compound, etc. But it's not really important to know now. You will understand it better when you're doing a pathology of the skin. But why this is important for us is a lot of these benign lesions could actually be cancerous, but most of these lesions of melanin pigmentation don't really mean anything, right? But if it's supposed to, if one of these lesions undergo a change in its genotype and has, basically some sort of mutation triggers its rapid production of the melanocytes, it could lead to melanoma, which is a really dangerous condition. Okay, so these are the last two pigments we're going to speak about for today. So we have lipofuscin. So lipofuscin is an age-related pigment which accumulates in cells which will not undergo mitosis anymore. And a term for this is known as brown atrophy because when you look at the organ of, say if you look at the heart of a really old person, say like an 80-year-old patient during an autopsy, you would see that their heart is slightly it's slightly brown tinted. So this is why it's called brown atrophy. And lipofuscin is a rather physiological pigment, which is there in most postmitotic cells. But ceroid, on the other hand, by the way, ceroid and lipofuscin look really similar, and you can't really differentiate it without the help of special stains. But yeah, so ceroid is also a type of lipopigment which accumulates in cells under some sort of pathological conditions. So, what are some examples of these pathological conditions? It could be due to some sort of destruction of adipose tissue, erythrocyte destruction, it could be due to the lack of vitamin E. It's also seen in a condition called melanosis coli and Dubin-Johnson syndrome. So Dubin-Johnson is a genetic syndrome. Yeah, so I guess we have to close off for today because the video is already really long, but we've not yet discussed pigments fully in detail. We've only touched upon the basics. We've just touched upon autogenous pigments. We're yet to discuss hematogenous pigments and exogenous. Then we will discuss a bit of foreign bodies, crystals, and concrements. And we will also move on to pathological calcification. So I will probably cover that in the next video. So see you guys. Bye bye.